My name is Joseph Georges. I am a PGY7 neurosurgery resident at the Philadelphia College of Osteopathic Medicine. And I would like to thank the Pennsylvania Neurosurgical Society for selecting our talk for presentation this year at the conference. The title of our talk is Provision of Rapid and Specific Ex Vivo Diagnosis of Central Nervous System Lymphoma from Rodent Xenograft Biopsies by a Fluorescent Aptamer. Prior to residency, I spent many years working in a neuroscience laboratory where I focused on live cell imaging and specifically studying the motility of glioma cells. This is imaging of glioma in a culture dish and just showing its mobility over a time course of about an hour gives us appreciation of how infiltrative and mobile these tumor cells are. In residency, I saw that when we scheduled patients for stereotactic biopsy, especially for small, deep-seated lesions, we could sometimes have trouble obtaining diagnostic tissue, and asked the basic question if fluorescence imaging techniques could help us improve the diagnostic yield for these small, deep-seated lesions. To answer that question, we started developing experiments at the Philadelphia College of Osteopathic Medicine, where we built small fluorescent microendoscopes and coupled them with stereotactic biopsy needles. And we developed animal brain tumor models where we in vivo would image regions of tumor prior to biopsy. Animals would be administered agents such as fluorescein, and after doing so, we found that in vivo we could visualize regions of tumor as compared to non-neoplastic regions consistently, and this would allow us to target tumor during, or specifically target tumor during the time of biopsy in these animal models. Fluorescein is great for labeling regions that enhance with gadolinium, but we know that that differential can be large. So we looked to see if there was a more specific available agent, and that brought us to 5-ALA, which is currently approved for use of resection during glioma in the United States. But we know through studies in our lab looking at other lesions such as metastasis and lymphoma that it could equally label other tumor types and allow us to quickly verify whether or not tissue we are sampling is tumor or not. We set up experiments looking at this agent where we took biopsies implanted with various tumor types. This one in specific is a breast cancer metastasis. And under room lighting, you can see biopsies from various regions can look similar. But if you administer 5-ALA and then look at these biopsies under a fluorescent light source, you can see immediately whether or not the biopsy was taken from a region of tumor versus a region not containing tumor. However, sometimes just identifying tumor or not is not enough. We need more specific information, the kind of information we obtain from antibody staining with special stains. And here's an example we see in neurosurgery. The frozen section on the right is that of a small cell astrocytoma, whereas the biopsy on the left is CNS lymphoma. And we all know that these two lesions have very different surgical plans, whereas one is typically operative and the other one is not. And being able to differentiate these is important during surgery. So we turned to fluorescence again. I knew of an agent known as sulfurotamine 101, which we used in the laboratory to quickly identify astrocytes during live cell experiments. And the basic question we had was, could it also quickly identify gliomas? And if doing so, could it do it in a time frame that could help with intraoperative decision making? So to answer that question, we set up experiments where we developed animal models of human glioma implanted with tumors. And we obtained biopsies from these rodents, incubated them with sulfurotamine 101, and found that it quickly labeled both glioma cells and reactive astrocytes in these tumors in a time frame of under 10 minutes. We also studied this agent in fresh biopsies from the operating room, found that it quickly labeled gliomas, whereas other lesions like adenomas, meningiomas, schwannomas, oligodendrogliomas, and specifically lymphoma, it provided negative staining. So sulfurotamine gave us a rapid means for identifying gliomas. We next looked to develop an agent that would allow us to visualize lymphoma in intraoperative time course. One did not exist, but we searched the literature and found a class of molecular probes known as aptamers that could quickly bind targets with antibody-like specificity, but they could be modified to undergo a conformational change when binding. We exploited this change to make them change fluorescence after binding. So when their target is not present, they fluoresce in one wavelength, whereas when they bind their target, they change in fluorescence. And we felt having a switchable agent specific to lymphoma would minimize staining steps and help increase the speed of diagnosis. We next studied this agent on xenografts implanted with CNS lymphoma. This is a slice of rodent brain containing a lymphoma modified to express red fluorescent protein as a positive control. We incubated these with our aptamer and found that it quickly labeled regions of tumor in a pattern similar to CD20 antibody staining, whereas in tissue not containing tumor, it did not label the cell bodies located in those regions. We quantified our data with a negative control of astrocytoma and found that our aptamer labeled approximately 80% of tumor cells with a false positive rate of under 10%. This particular switchable agent took about 45 minutes to 60 minutes for a diagnosis, which is too slow for intraoperative feedback. So we next truncated this aptamer and further modified it for more rapid staining. We did a time course looking at 5, 10, 20-minute staining in various concentrations and found that within 10 minutes of biopsy, this agent could give us a positive diagnosis of lymphoma from rodent biopsies. This is looking at our agent with a negative control of glioma, and we found minimal to no staining from negative control tissues, including glioma. So these projects have brought us to a point where sometimes we see these small deep-seated lesions that we plan for stereotactic biopsy, and we do run the risk of having non-diagnostic tissue. With fluorescein, it can help us in vivo identify neoplastic tissue prior to biopsy. With 5-ALA, we can know immediately in the operating room if the lesion we're biopsying is tumor versus not tumor. And then if we needed more specific diagnosis, we can use agents such as sulforhodamine 101 that within 10 minutes or so can identify astrocytomas. And we now have our truncated aptamer, which within 10 minutes of biopsy can identify human B-cell lymphoma from rodent biopsies. We are next developing clinical studies where we are going to recruit more patients and study these agents on fresh human biopsies. I want to thank the funding sources for these projects, which include the NIH, the Philadelphia College of Osteopathic Medicine, and the Barrow Neuroscience Foundation. I also would like to thank my mentors and collaborators, specifically those at the Philadelphia College of Osteopathic Medicine, who have helped me continue these projects and provide more mentorship as we've developed additional techniques for fluorescence in the operating room. Thank you.